into the important news of the day. For public affairs television that expands your vision of the world, turn to C-SPAN, open to the public.
Coming up next, a town forum entitled Meeting the AIDS Challenge in the 90s. The discussion is sponsored by the Institute for Advanced Studies in Immunology and Aging and is moderated by syndicated columnist Jack Anderson. Participants include Dr. Jonas Salk, who developed the polio vaccine and has developed an AIDS vaccine, Dr. Robert Gallo, the scientist who discovered the AIDS virus, and representatives of several AIDS advocacy organizations.
I'm very concerned about the AIDS problem.
These are my two concerns right here. They're the ones that have to deal with it right now more than I do.
It's affected some of my friends. I have a couple of friends who had it, died from it, so it scares me to death.
Well, I think more money should be given to research and finding a cure for it. And well, I've had my own AIDS tests, and I think that's really, 10 years ago I wouldn't have thought about anything like that ever happening, where I'd be put in a position where I have to worry about my life like that.
I have lots of friends who I don't know from day to day, or they don't know from day to day what's going on.
It's sad. We need more funding. There has to be more done.
More education, too.
More education.
To understand it.
It's a big problem, and I am very concerned about it. People still are afraid that if you shake somebody's hand, you're going to get it.
I've known people that have died of it, and I think it's terrible. I think it's a tragedy. It's horrible.
We feel so helpless in this crisis. I think a lot of people do. Anybody in the healthcare profession feels very helpless.
Ladies and gentlemen, thank you and welcome to a town forum on AIDS. Meeting the AIDS challenge in the 90s. I would like to take this opportunity to thank our host, the George Washington University, and share with you a little bit about the work of the Institute. The Institute includes funding research through fellowships, bringing scientists from around the world here to George Washington University to provide educational opportunities such as today's forum. The crisis that is before us is well known to everyone in this audience. I'd like to share with you for a moment a brief video about the Institute for Advanced Studies in Immunology and Aging.
An education organization. Since 1985, the Institute has been a driving force in the research being conducted on the body's complex immune system. The Institute's major focus is on defining the linkage between the immune system, the brain, and aging, and accelerating the prevention and treatment of major diseases through the use of thymosins. Educating the public on the latest advances in the areas of immunology and aging is a priority, along with providing a collaborative setting for scientists to share vital information.
My group, basically, has been involved in looking at two different areas. They overlap, but they're really two distinct areas, both of which have been supported to a large extent by the Institute, particularly through its visiting fellows programs.
Primary activity of the Institute is the awarding of scholarships and fellowships to promising young scientists, allowing them the extraordinary experience of working on the cutting edge of biomedical research.
My work here is involved in trying to define the interactions between the brain and the thymus gland. Hopefully, through the work I'm doing, we'll be able to define the interactions and be able to resolve the processes involved and perhaps treat or prevent some of the conditions that are seen, such as Alzheimer's, cancers, other immune defects.
Essentially, my research here at the Institute is concerned with the optimization of our candidate AIDS vaccine, HGP-30. I'd like to thank the Institute for providing me with a fellowship for funding my graduate research. When I decided a few years ago to go to graduate school and pursue my PhD, the one inflexible requirement that I had was that my research be concerned with an AIDS-related project, and so I'm very grateful to the Institute for providing me with the opportunity both to pursue my PhD and also to do so in a manner that I find very important.
The Institute's ambitious health communications program supports a renowned series of scientific meetings and open public discussions to further its primary mission, accelerating the pace of scientific discovery to improve the quality of life.
Ultimately, honoring individuals that are contributing to the support of research and all of this equipment. Ultimately, it's about saving lives and improving the quality of health to try and deal with these terrible afflictions, and through the support of the community and friends, we hope to accomplish something that will translate into not only saving lives of children, but saving lives of adults, our parents, our grandparents, our friends and neighbors through the work of the Institute. And if we can save one life through the work of the Institute, it will be worthwhile.
I'd like to introduce our host for today's meeting, Mr. Stephen Trachtenberg.
Thank you very much. It is with sadness, with pride, and with concern that I welcome all of you to this town forum and the update it will offer on recent and current advances being made in AIDS research and treatment. Sadness at the thought of how many unknowns still cluster around our understanding of this tragic affliction. A tragic affliction which has already taken so many lives in our community, in our country, and around the globe. Pride that a meeting of such importance with such very interesting panelists is being held at the George Washington University under the guidance of my colleague, Dr. Alan Goldstein. Concern that this town forum prove successful in helping to sustain the momentum of the research efforts now being made towards a more sophisticated knowledge of the immune system in general and its relation to AIDS in particular. I'd like to offer a brief reflection from the years of my own childhood, a reflection prompted by the association of this initiative with Dr. Jonas E. Salk, whom I and others of my generation long ago learned to associate with the archangels and other major protectors of our vulnerable and fallible species. To an extent difficult to imagine today, my generation grew up with many versions of a single picture in front of our eyes. Photographs and drawings of the old iron lung that was the only way of dealing with infantile paralysis, polio, until Dr. Salk developed the vaccine that will always be associated with his name. Those pictured varied very little. There was the sheer dark immensity of the lung itself. There was the diminutive head of a young boy or girl emerging from one end of it with a slanted mirror above his or her face so that objects or pages held in front of the mirror could be seen in a reasonably normal way. And there were usually 2 or 3 concerned adults in the photo as well, to soften the awareness that the boy or girl was alone inside that machine 24 hours a day, an aloneness mainly punctuated by the visits of nurses and other medical caretakers. In those days, public swimming pools, having become identified as major sources of contamination, and public swimming pools playing a role of such importance in an age before suburbia and air conditioning, few parents watched their children march off with their towels and their bathing suits without at least a touch of apprehension. They and their children were taking a calculated risk. There might be an iron lung in their future. I recall that time so utterly transformed by Dr. Salk's great discovery to place just a bit of perspective around the AIDS challenge we face today. A past major peril affecting the entire species was brought under control. In subsequent decades, the ancient scourge of smallpox was tracked down, continent by continent and nation by nation. It too was brought under control. Specimens of the virus artificially preserved just in case. Measles, never the routine, trouble-free illness that some people imagined, has moved some distance towards a similar fate. But the recent upsurge of measles in this country reminds us of what can happen when medical advances are not pursued with the same dedication and determined spirit that human beings have so often shown in time of war. And that's why today's gathering here at George Washington University is of such tremendous importance. Scientists may pursue their researches with unflagging zeal, the way Dr. Salk did when he developed his vaccine. But in today's world, which offers so many competing agendas having to do with the expenditure of tax dollars, public understanding and support of such research is necessary to an extent that makes unprecedented an appropriate adjective. On behalf of the George Washington University, I greet you. On behalf of the human race, I thank you. May your discussions and the exchanges reach the ears that most need to hear them. Good luck. Thank you very much.
Thank you, Mr. Trachtenberg. My name is Rick Hinden. I am chairman of the board of a nonprofit organization called the Institute of Advanced Studies in Immunology and Aging. We are here today as part of our philosophical mission to educate and share the advances in science with the community. It was our thought that while much of this information is understood on a scientific level, that it needed to be brought to the public at large. So it is with that effort that we, with the help of so many volunteers, put together today's forum. The event will go as follows. We will first have a 5 or 6 minute discussion by each of our renowned scientists on their particular area of expertise in the science. Then we will have a question and answer period by first a scientific journalist panel, and secondly an AIDS activist panel. And thirdly, and probably why many of you are here today, is to answer questions to the public at large. You will have an opportunity to ask those questions. I'd like to make just one statement and one thought clear. The Institute is not a political organization, nor are we trying to make any social statement. We are simply here for one purpose, to share the latest information on the science and research surrounding AIDS. With that, I would like to introduce our moderator. Jack Anderson is one of America's foremost investigative reporters. Mr. Anderson is nationally syndicated. His column, Washington Merry-Go-Round, is distributed to some 1,000 newspapers by United Feature Syndicate. His relentless digging and to the deceit, corruption, and waste at the highest levels of government has earned Mr. Anderson the respect and praise of both his readers and his fellow journalists, as well as a Pulitzer Prize in 1972. Ladies and gentlemen, please join me in welcoming Mr. Jack Anderson.
Thank you. I listened to the introduction with grateful embarrassment. I feel a little like the British nobleman who nudged his neighbor and said, "I had a horrible dream last night. I dreamt I was addressing the House of Lords, and when I woke up, by Jove, I was." I find doctors delightful at my bedside, a little formidable in one group, and I should confess right at the outset that I am a medical illiterate. I'm not quite sure why I was selected for this honor. I will say to you that we live in an exciting and exhilarating time, that the future is bearing down on us like an onrushing express train, that is very likely to arrive here before we're ready. The world doubled its knowledge in the last 3 years. The more we learn, the faster we learn. The faster we learn, the more we learn. And so meetings like this, information such as you're going to hear today, the participation of the experts who are keeping a step ahead of the game, these are vital to our progress, to our national progress. I'm honored to be part of it. But because of my own medical illiteracy, the staff here at George Washington University has mercifully prepared a script. And my biggest struggle will be just to pronounce everything correctly, because people here can spell better than I can pronounce. So now, ladies and gentlemen, please welcome the members of our press panel, Ms. Barbara Culleton of Science, Ms. Ann Giudici-Fettner of Medical World News, Dr. Susan Oki of the Washington Post, and Mr. Alfred Rosenfeld from Omni Longevity Magazine. And now please welcome the members of our advocacy panel. Dr. Peter Hawley, the medical director of the Whitman Walker Clinic. Ms. Sandra McDonald, chairperson of the National Minority AIDS Council. Mr. Michael Murdian, executive director of the National Association of People with AIDS. And Mr. B.J. Stiles, president of the National Leadership Coalition on AIDS. And now it's my distinct pleasure to introduce to you our esteemed scientific panel. Dr. Anthony Fauci is the distinguished immunologist heading the U.S. government's war on AIDS. Dr. Fauci has pioneered the field of human immunoregulation by making a number of basic scientific observations that serve as the basis for current understanding of the regulation of the human immune response. In addition, Dr. Fauci is widely recognized for delineating the precise mechanisms whereby immunosuppressive agents modulate the human immune response. Most recently, Dr. Fauci demonstrated the precise nature of the immune defect in AIDS and has been instrumental in developing strategies for the therapy and immune reconstitution of patients with this serious disease. He continues to devote much of his time to identifying the nature of the immunopathogenic mechanisms of AIDS and the scope of the body's immune response to the AIDS retrovirus. Dr. Fauci is director of the National Institutes of Allergy and Infectious Diseases of the National Institutes of Health. He received his medical degree from Cornell University Medical College in 1966 and became director of NIAID in 1984. He also serves as associate director of NIH for research on the acquired immunodeficiency syndrome and as director of the NIH Office on AIDS Research. Dr. Fauci is the recipient of numerous prestigious awards for his scientific accomplishments and is a member of the leading professional societies, including the Institute of Medicine of the National Academy of Sciences. He's also on the editorial boards of many scientific journals and is co-author or editor of more than 650 scientific publications, including several textbooks. Ladies and gentlemen, Dr. Anthony Fauci. Dr. Robert Gallo is the distinguished American scientist who, with his co-workers at the NIH and with scientists at the Pasteur Institute, is credited with discovering the AIDS virus. He's universally recognized for his achievements in molecular biology and for his pioneering studies in the field of human retrovirology. His research led to the rapid development of the ELISA blood test to detect the AIDS virus, which is already saving thousands of lives. The discovery of the human T-cell growth factor from his laboratory in 1976, and the development of novel technology to grow T-cells in continuous culture has revolutionized modern immunology and was the major technological advance that enabled human retroviruses to be grown in tissue culture. Most recently, Dr. Gallo and his co-workers have made important advances in our understanding of the pathogenesis of Kaposi's sarcoma, a cancer frequently associated with AIDS, by discovering that TAT, an HIV viral regulatory protein, can escape from HIV-infected cells and can directly stimulate the growth of Kaposi's cells. This work provides a valuable focus for researchers attempting to develop drugs against this cancer-like proliferation of blood vessels and skin cells, which has become a hallmark of AIDS. Dr. Gallo is a native of Waterbury, Connecticut, and received his B.A. summa cum laude in 1959 from Providence College and his M.D. from Jefferson Medical College in 1963. He's been head of the Laboratory of Tumor Cell Biology at the National Cancer Institute of the NIH since 1969 and has published over 800 scientific papers, most of them on the subject of retroviruses. Dr. Gallo, who was elected to the National Academy of Sciences in 1988, has received numerous national and international awards and honorary doctorates from universities in the United States, Italy, Israel, and Belgium. It is a unique circumstance that he's been awarded the Albert Lasker Award in Medicine on 2 occasions. Ladies and gentlemen, Dr. Robert Gallo. Now, Dr. Ari Rubenstein is a distinguished pediatrician and a leading authority on the treatment of children with AIDS. His pioneering studies involve passive immunotherapy, biological response modifiers, and antiviral therapies have provided new insights into the development of novel treatment modalities which have greatly improved the quality of life for children with AIDS. For the past several years, Dr. Rubenstein's research has focused on identifying specific antibodies that can prevent the transmission of AIDS. These studies have led to the development of a novel prototype GP120 envelope-based vaccine with a primary neutralizing domain designed to block the transmission of the AIDS virus from an infected mother to the developing fetus. Dr. Rubenstein is director of the Center for AIDS Research at the Albert Einstein College of Medicine and is an internationally renowned authority in pediatric immunology. He sees more children with AIDS than anyone in the world and has pioneered in their treatment. Dr. Rubenstein received his medical degree at the University of Bern, Switzerland, and postdoctoral training at the University of Tel Aviv, the University of Bern, and Harvard Medical School. He's currently professor of pediatrics and microbiology and immunology at the Albert Einstein College of Medicine. He's on the editorial board of the Journal for Clinical Investigation, the Journal of Pediatrics, AIDS, and a number of other important medical journals. Ladies and gentlemen, Dr. Ari Rubenstein. Dr. Jonas Salk is a distinguished virologist and immunologist, best known for his pioneering work, which led to the development of the first effective polio vaccine. Most recently, he has developed a novel, killed, whole virus, envelope-less AIDS vaccine currently being tested in human trials. He's the author of more than 100 scientific papers and several books, including Man Unfolding, The Survival of the Wisest, World Population and Human Values, A New Reality, and Anatomy of Reality, Merging of Institution and Reason. Dr. Salk has been the Distinguished Professor of International Health Sciences of the Salk Institute for Biological Studies since 1984. He's been the founding director of the Salk Institute since 1963 and was a resident fellow until 1984. Since 1970, he's been an adjunct professor of health sciences at the University of California, San Diego. Dr. Salk has received numerous honors and awards, including a presidential citation, the Lasker Award of the American Public Health Association, a Congressional Gold Medal, and a Presidential Medal of Freedom. He's a fellow of the American Academy of the Advancement of Science and the American Public Health Association, an associate fellow of the American Academy of Pediatrics, and a member of numerous other professional associations. Dr. Jonas Salk. Dr. Alan Goldstein is one of the leading American authorities on the thymus gland and the workings of the immune system. He is co-discoverer of the thymosins, the hormones of the thymus that control immunity, and the discoverer of a novel P17 AIDS vaccine, HPD-30, that is currently undergoing human trials in London and has just received approval from the State of California Food and Drug Board to initiate trials in San Francisco and Los Angeles. Since 1978, Dr. Goldstein has been chairman of the Department of Biochemistry and Molecular Biology here at George Washington University School of Medicine. He has served as consultant to many research organizations in industry and government, including the Division of Cancer Treatment, National Cancer Institute of the National Institutes of Health. As the scientific director of the Institute for Advanced Studies in Immunology and Aging, Dr. Goldstein has worked extensively in the areas of immunology, aging, AIDS, and cancer. Ladies and gentlemen, it is my pleasure to introduce the convener of today's forum, Dr. Alan Goldstein.
Thank you, Mr. Anderson. This is George Washington University. My son goes to Georgetown Day School, but this is George Washington University. I would also like to welcome our distinguished panelists and guests to our University and Medical Center. This is the first in what we hope will be a series of informative and educational town forums sponsored by the Institute for Advanced Studies in Immunology and Aging. Surprisingly, in areas dealing with basic biomedical research, there are very few town forums in Washington that the public can get involved in the way they are today. I believe that what you will hear today will suggest that as we look to the decade of the 90s, although we have many questions to answer and problems ahead, there is a ray of hope. Just at a time when our health experts were predicting the eradication of the great infectious epidemics of our time, such as smallpox and polio, AIDS entered the picture with vengeance and was brought to our attention not by epidemiologists or health officials from third world countries, but by uptown physicians working in some of the major centers of America. From the beginning in 1980 and 81, the story of the AIDS epidemic began to unfold. It was originally thought to be a gay disease, which it is not. Then it was thought to be an American disease, which it is not. AIDS is now recognized to be a public health threat worldwide. Estimates from the World Health Organization of 5 to 10 million infected would include perhaps as many as 1.5 million Americans. To date, over 130,000 Americans have AIDS, and about half have died. By the end of 1981 and early 82, it was clear to many scientists and clinicians on the front lines that this was not the garden variety disease of the month, but rather a major public health problem that, if not contained, could be devastating. We were at war against an enemy whose face we did not know, that was causing massive destruction of the immune system by mechanisms that were not clear, and that the only means we had to treat this terrible disease was by using old weapons such as antibiotics that were only useful in dealing with the secondary opportunistic infections that were attacking individuals with this strange affliction. The pleas of the research community for funding in the early 80s to support new research initiatives went largely unheeded. We did, however, have some very positive weapons of our own that enabled us to rapidly identify the face of the enemy. Thanks to the pioneering work and creativity of individuals such as Dr. Robert Gallo and his colleagues at the NIH, along with scientists at the Pasteur Institute, by 1984 we realized that the face of the enemy belonged to an RNA virus that has been termed HIV. With the information that the virus could be transmitted only by sexual contact or by blood transfusions or by infected needles, scientists at the NIH very rapidly developed a blood test called an ELISA test, which could protect our blood supply and is already saving thousands of lives. Furthermore, thanks to previous advances in molecular biology, genetics, and immunology, we could very quickly define the mechanism by which the virus entered the cell and is integrated into the nucleus and is activated, as well as the immune mechanisms necessary for developing approaches to contain the threat of the virus. You will hear about some of this remarkable progress this afternoon from Dr. Gallo and Dr. Fauci. We are also making significant progress in the vaccine front. We are delighted to have with us Dr. Jonas Salk, the father of the polio vaccine, who is helping to guide the development of the killed whole virus core vaccine that is showing significant promise in HIV-positive individuals. You will hear from Dr. Ari Rubenstein on the development of a new prototype vaccine that is being developed from the envelope of the AIDS virus, designed to protect the mother from transmitting the AIDS virus to the developing fetus. I'm also pleased to report that here at the George Washington University Medical Center, we are also making progress. In my own group, Dr. Paul Naylor and a team of young research scientists, such as some of the young ladies that you saw in the video, have since 1985 been working on development of a synthetic subunit AIDS vaccine whose active component is a highly conserved region of one of the inner core proteins of the AIDS virus called P17. These studies, which have been carried out in collaboration with Dr. Prem Saran at the NCI and Dr. Joe Gibbs at the NIH, and with Dr. James Rubin and Don Rios in Houston, have resulted in the development of a prototype vaccine termed HGP-30. One of the major hurdles in developing an effective AIDS vaccine is the problem that the outer coat or envelope of the virus keeps changing by a process called genetic drift. This genetic drift provides a moving target for the immune system, which is difficult to overcome. We think that the vaccine HGP-30 may overcome this problem, as the active component is a highly conserved region of a core protein, which is essentially the same in all variants of the AIDS virus. The first human testing of HGP-30 in healthy HIV-negative individuals is nearing completion at the Kobla AIDS Center of St. Stephen's Hospital in London, and the results of this phase 1 study will be presented at the International AIDS meeting in San Francisco in June. Over the past 10 years, I've been a witness to the unfolding of this AIDS epidemic, and I am quite encouraged by the progress that has been made that you will be hearing about this afternoon. We hopefully will be able to translate this progress into an effective treatment and cure for this deadly disease in this decade. We should also keep in mind that what we are doing today in this fight against AIDS will help prepare us to meet any new unexpected challenge that may come our way in the future. As we have learned from previous experiences with epidemics, that as long as life exists on this planet, we will need to keep a constant vigil against new microbes that will surely evolve to test the will and fiber of our society. How effectively we come together as a nation to meet this challenge will say a lot about the health of our nation and our hope for the future. Thank you.
Thank you, Dr. Goldstein. I apologize for that Georgetown slip. For a moment, I got basketball and medicine mixed up with it. Our next expert is Dr. Anthony Fauci. Dr. Fauci.
Thank you very much, Mr. Anderson. Dr. Goldstein has asked me in 5 minutes to not only review the research that I've been doing for the last 22 years, but to tell you all about what the NIH has done in the last 9 years in HIV research. So without any further ado, I'll get to the task. Actually, I think an outline of what is going on at the NIH will be covered in the discussions that we'll have with the various panels, but let me very briefly outline the topics of research that is being conducted at the NIH and elsewhere, but for the purposes of the discussion now, is the research on the natural history and the epidemiology of HIV. And just a statement there, many of you have heard that we are seeing a changing face of the epidemic of HIV, because we're simultaneously having an epidemic of infection at the same time you have an epidemic of disease, except that they're separated by about 8 to 10 years. So the people that are getting infected now are going to be the population predominantly that are going to have disease 7, 8, 9, 10 years from now. And the people who are getting disease now are the people who were infected years ago. So when you talk about decreasing rates of infection, for example, among gay men, that doesn't mean you're not going to see gay men who have disease for the next several years. And when you talk about the now clustering of new infection in the inner cities, particularly among minorities, IV drug users, pockets that have already been underserved by the healthcare system, that's something that's going to be even worse in the 1990s. The next area is the area of the etiology, HIV and related retroviruses. You'll hear a bit about that from Bob Gallo. I'm going to talk in a moment, just for a minute or so, about the research conducted in my own laboratory, namely pathogenic mechanisms of HIV. And then the two final areas that are of great importance, obviously, to the public in general, are the area of treatment. And in the 1990s, we will be seeing some revolutionizing of the way we look at treatment of HIV, because the end of the 1980s has created the concept that you'll see in the 90s of early intervention, namely treating people early on before they develop full-blown disease, prophylaxing them against opportunistic infections, to hopefully have the goal of the decade of the 90s to convert HIV infection into a chronic manageable infection, whereby you can test someone, counsel them, get them into the care of a physician, and treat them with a combination of drugs early enough on in the course of infection so that you might have a situation like many other chronic diseases where there's the feasibility of a reasonable, comfortable lifespan. And then finally, the area of vaccine, which you'll hear a bit about from Dr. Salk and some also from Dr. Rubenstein. And again, 1989 was a good year in this regard, not saying that we're going to have a vaccine soon, but the concept of what we call protective immunity in animal models and in humans has really begun to be established. So a year ago, if I were addressing you, I wouldn't be able to say whether or not we were going to have a vaccine. I think most scientists now are reasonably optimistic that sometime, hopefully in the 1990s, we'll have a vaccine based on some of the things you'll hear about. Now, for the last couple of minutes, let me just outline for you the area of research that I have been conducting with my colleagues in my laboratory over the past several years. Prior to the HIV epidemic, we were concentrating on the regulation of the immune system. And if it wasn't for the decades of knowledge that was accumulated on the immune system, on molecular biology, on microbiology, certainly we would not have been able to address the way we have been, we being the scientific community, the HIV epidemic. If it had happened in 1950, we would have still been wondering what was going on. The area that we've concentrated on is how this virus, in the most unusual way, has incorporated itself into the body's immune system in what we can say is a very smart and very diabolical way. It uses the receptors on cells of the immune system that are functionally important cells to gain entry into the cell. It uses a variety of immune-mediated mechanisms to cause damage to the cell. But the area that I had been most interested in is answering the question, if you have someone infected with HIV and the virus can lay latent or dormant in their cells, why is it that over a period of 8 to 10 years there's a gradual, insidious, but invariable diminution of their immune function, where the virus sooner or later wears out the body's immune system, which translates to a mechanistic question of what is it that induces this virus to come out of the latent state into a productive state, whereby it destroys the cells of your body that are responsible for protecting you, namely your immune competent cells. And the hallmark and the common denominator of HIV infection and AIDS is a destruction of those cells, leaving the body prey to opportunistic diseases, be they infections or cancers. And we've been able to show over the past few years that the very regulatory molecules that are responsible for causing a homeostatic regulatory state of the immune system are the molecules which can actually upregulate virus expression. So you don't need a dramatic event, another infection, or some sort of massive stimulation of the immune system. What the virus has done, again, if you want to personify it, has recognized that if it intersperses itself into the immune system, it can use those very mechanisms, those factors that cells use to talk to each other. That's what's called immunopathogenesis. That's what we have been working on. That's what a number of laboratories throughout the world has been working on. It's more than of just academic interest, because if you can identify at the cellular and at the molecular level these mechanisms, then you have a scientific basis for interrupting these mechanisms. And that's exactly what you're going to be seeing and reading about in the 1990s. This is a specific targeted interruption of these cellular and molecular mechanisms. And let me just close by saying I alluded to the fact that if it had not been for the fundamental basic knowledge in immunology, we would not have been able to appreciate all of the pathogenic mechanisms of HIV infection. We still haven't appreciated them all, but we certainly have made advances. And as is always the case with basic science, the information that we're learning about the immune system and about microbiology and molecular biology from studying AIDS is going to give us great insight into diseases a decade or two from now that have absolutely nothing at all to do with HIV infection. Thank you.
Thank you, Dr. Fauci. Now our next distinguished panelist is Dr. Robert Gallo.
Well, I'll say some things that parallel Dr. Fauci. My coworkers and I believe that perhaps the greatest help we can be in the AIDS problem is by focusing on basic mechanisms and how the virus works. How does it produce the disease we call AIDS? How does the virus successfully infect the cell in detail? That is, looking at each step of the virus replication cycle at what we call the cellular and at the molecular level. An example would be that the field now knows, and has known for some years, that to enter a target cell in general, the AIDS virus needs to stick onto, through its envelope, the molecule called CD4, and that a way to block that would be to use pure CD4 molecules as a competitive inhibitor with the cell. And that's an avenue of research conducted now in several pharmaceutical laboratories. We know that the next step in the virus infectious cycle is the, if you'll allow me to speak loosely, the conversion of the virus's genetic information in the form of RNA into DNA. It's known from the beginning that this is catalyzed by the special enzyme reverse transcriptase. This is where AZT works, DDI, DDC, and perhaps it's an area of research that needs more study wider. That is, there are all kinds of ways to inhibit reverse transcriptase. The focus is on one or two right at the present time. And this is an interesting target because it's specific. It's an enzyme that makes DNA, therefore we call it, as many of you know, a DNA polymerase. And the cell has DNA polymerases. It has at least 3. But the viral DNA polymerase, reverse transcriptase, has some important differences from the cellular enzyme, and therefore becomes a potential target. You all know that a virus is much more difficult to interfere with than a bacterium. That's why antibiotics don't work, because a virus doesn't have a metabolism of its own. And drug companies in the past got away from research in antiviral agents, more or less. AIDS has brought them back. In fact, whatever the legacy of AZT is, for sure it's done one thing, and that's to make pharmaceutical companies pay attention to the possibility of antiviral therapy in AIDS. There are enough specific components to the virus, enough specific steps in the virus's stage-wise process of reproducing itself that one can suggest, at least from the basic studies, ways to interfere. After it makes DNA from the RNA, that DNA has to travel. It has to get to the nucleus of the cell, and it has to then insert itself into the DNA of the normal cell, into the chromosomal DNA, the process called integration. We know that this process is rather specific. It requires subtle molecular mechanisms. They too could be targets. They are not right at this moment. That is, there's little study at that level. Once integration occurs, Dr. Fauci talked about a quasi-latent period. I don't think, and I think he doesn't think, that the disease has a real latency. There's always some cells expressing virus, but we can talk about latency at the cellular level. That means that any one time, many cells can harbor the viral genes integrated, but they don't express those genes. The genes are silent. They're not making viral RNA again and viral proteins and reforming virus. He talked about things that turn that process on. But once the process is turned on, that cell is doomed to die if it's a T4 lymphocyte, once the virus begins to form. And the stages of turning on to the formation of the virus are multiple. The integrated DNA has to remake the viral RNA. That requires some specific steps. And we know for the AIDS virus, the 2 regulatory genes, known as TAT, TAT, and REV, are critical to those stages in the nucleus of the cell. As such, they become important targets of future chemotherapy because the cell doesn't have exactly those genes. I'm sure we have related genes that do related things, and from the AIDS virus we're learning about normal cell biology all the time. And we would assume that since a virus has to usurp a cell's function, that these genes have put themselves into a pathway that the cell normally uses but hadn't been discovered before, that out of AIDS research has come that discovery. But in any case, these two genes, Rev and TAT, are absolutely specific to the AIDS virus. They can be targeted already by a technique called antisense RNA approaches. That kind of work is going on in our laboratory now. But it's a new area of chemotherapy, very new, and it's hard to predict. And it would be foolish for me to say in this year or that year we can promise this or that. There's still more steps that are viral specific that give hope. For example, and I'll mention one last one, after the virus's RNA is formed in various ways and it's in its mature state, the virus can't form unless viral proteins are also made. Some of the viral RNA acts as a messenger to be translated, to make viral proteins. The viral proteins are usually made large. They have to be cut. They're cut by specific enzymes known as proteases. Sometimes it's a protease of the cell that does it. But for a major core protein, one of them that Dr. Goldstein referred to, the precursor of that protein has to be cut to smaller size by a specific protease that the virus carries. This is an important target for chemotherapy of the future, and currently it is being approached, attacked, whatever you want to call it, by many pharmaceutical companies. I could think of at least 4, maybe more, and there are some interesting results that are steadily coming out. So antibiotics, in quotes, for a virus has to be far more sophisticated than it does for a bacterium or a parasite, and it brings in a new era, and I think out of AIDS that will occur. I'll mention also that we believe that, I said that perhaps our best help to this field can come from the basic studies of how the virus works to reproduce itself and to cause disease, but we also feel that to do these studies requires systems. And that may be obvious. You can't get the answers unless you have a way of studying it. Well, sometimes systems to ask and answer a question do not exist. Sometimes it's difficult to go find them and make them. But without finding them and making them, you can't advance very well, except by almost blind clinical trials. We felt this way strongly about Kaposi sarcoma. And so in the last few years, we've developed systems in the laboratory, in animal and in tissue culture, to study Kaposi sarcoma. We think this has led to some interesting results that Alan kindly referred to, or Mr. Anderson, I guess, did. The TAT protein of the virus actually can make the apparent precursor cell of Kaposi sarcoma grow. This was a surprise. The TAT protein is normally working to regulate the expression of the virus. But it turns out that an infected T cell can liberate TAT, and that it works on precursors of cells that become the dominant cell of Kaposi sarcoma as a growth factor. A really unexpected result. In this system, we can think of ways to try to interfere with the Kaposi sarcoma development. And we can test candidate compounds that come to our attention or that we happen to think about, to imagine, to fantasize about, and then to test. And we're testing some compounds in that way now in collaboration with some pharmaceutical companies. Lastly, we work modestly, maybe to a minor degree, in areas that Dr. Salk will talk about, immune therapy. We do this really only by collaboration with a colleague at the University of Paris, Daniel Zaguri. Our efforts here are similar to the efforts I just described to you, except they're more in the immune system. That is, we try to find out what component of the virus, what protein, what portion of what protein, what fragment of what protein induces this or that type of immune response. All of you know that there's immunity and there's immunity. The immune system is complex. You can stimulate it in many different ways. Sometimes you can make things worse. So we try at a basic level with many other laboratories to find out what protein does what and in what way to help people both for immune therapy in an infected individual and also to take part in a vaccine program. My belief is that a vaccine from AIDS will not be a single individual-like thing, such as Jonas Salk's with polio. It's already been a mountain of collaborations and will continue to be so with contributions from many areas because it's a far more complicated problem and a far more complex virus. Those are the current things that we're involved in now. Thank you.
Thank you, Dr. Gallo.
Our next expert is Dr. Ari Rubenstein. Dr. Rubenstein.
It's a great honor for me to be here with distinguished investigators like Dr. Fauci, Dr. Gallo, Dr. Salk, and Dr. Goldstein, who in fact were my teachers, our teachers, teachers of those who deal with patients with AIDS day in and day out. I am representing many investigators at the Albert Einstein College of Medicine. Our medical school is located in the midst of a borough of New York City that has a large inner city poverty area population, mainly minority populations. So that we encountered AIDS, we encountered it in a different way that most of the other centers did. The first patients that we have seen in 1978 and 1979 were children who presented with a very bizarre immune deficiency that was different from all other immune deficiencies that we have seen before. The most unique part of that immune deficiency was that not only the child was immunodeficient, but also often his mother or his father. And this led us to understand that we are dealing here with a so-called infectious immunodeficiency that later on turned to be due to the AIDS virus discovered by Dr. Gallo. We had to work hand in hand in the clinical field, in the psychosocial area, and in clinical research related to pediatric AIDS and AIDS in women and in families. And in fact, in 1981, we developed the first pediatric AIDS center, and when we realized that the child is not the only infected individual in the family, but it's a family disease, 2 years later, we have started with a comprehensive AIDS family care center. We have then developed also daycare centers, initially segregated daycare centers for children with AIDS, then integrated daycare centers, residential care facilities, recreational and educational camps for parents with AIDS, for children with AIDS, and for foster parents with AIDS. Research-wise, we have been involved in the identification of the clinical presentation of the disease in children, identifying two populations, as Dr. Fauci described to you before: those who develop the disease very early in life, in the first months of life, and die before they reach the age of 2 years, and those who live much longer. And some of our patients that we have identified in 1978 are still alive with us today. We have tried to identify the different immunoprotective mechanisms in those long-term survivors in order to be able to identify or to develop treatments for these patients. Some of the treatments that we have been intensely involved with were the use of intravenous gamma globulin for these children and hyperimmune serum for these children, namely intravenous gamma globulin that had activity, specific activity against the AIDS virus. But what was most intriguing for us was the epidemiology of the disease. We knew that the disease is in most instances passed from a pregnant HIV-infected woman to her fetus, to her baby. However, not all women who were HIV-infected transmitted their disease. Only about 30% to 40% transmitted the disease to their baby. So that 60 to 70% of HIV-infected women carried the virus. And we knew that this virus crossed the placenta. We could identify in the newborns, often maternal cells with the AIDS virus. And yet 70% of these babies were not infected by the AIDS virus or did not develop a disease with the AIDS virus. We have therefore recently looked at specific antibodies that can discriminate between women who transmit the disease and those who do not transmit the disease to their babies. And some of this work has been done collaboratively with many other investigators, including Dr. Devash from Ortho and previously from DuPont. We have finally identified an antibody, a so-called high-affinity antibody, that reacts with a small piece of the AIDS virus, of the glycoprotein 120 that Dr. Gallo mentioned to you before. And it turned out that women who had this high-affinity antibody to this small piece of the GP120, the piece that we call the primary neutralizing domain, or the PND, that those women who had these antibodies did not transmit their disease to their fetus. On the other hand, all women who did not have this high affinity antibody had babies who were HIV infected. And this finding led us to think about the development of a new form of a vaccine, the so-called PND vaccine, using this small piece of the GP120 and designating an approach by which women who cannot make the high affinity antibodies would be immunized by this PND vaccine, with the hope that they will then produce a high affinity antibody that, when they become pregnant, will protect their fetus from infection by the AIDS virus. This approach, which is currently only designated for HIV-infected women, may in the future also be used as a preventive vaccine, a transmission-blocking vaccine in HIV-uninfected individuals. Thank you.
Thank you, Dr. Rubenstein. And now our final distinguished panelist, Dr. Jonas Salk. I was supposed to turn the light on for you, Dr. Salk, but I can't figure it out.
Thank you very much. I accepted this invitation to come here today, hoping to contribute in a small way to the social enterprise that science represents, as you gather from the diverse approaches that have been described to you by my colleagues. When a few years ago, Bob Gallo and I talked about how one might approach dealing with this problem, it seemed to me that the strategies might be broadened beyond those that were then being employed. And I have brought with me a few slides just to give you some images to take back with you. Besides the words, I can just imagine you going home later and describing to your friends about the TAT gene and the proteins that are produced by it, and the GP120 molecule. And so I thought that I'd help you with a few pictures that will let you see what it was that I was looking at and what led me to the strategies that we've employed. And so in order to save some time, and perhaps we'll have questions that you might have asked of me, let me have the first slide, please. The first slide, if I remember correctly, describes in this pyramidal form the population of AIDS patients, of infected individuals in the world, and you see the large number of individuals who are HIV infected, are asymptomatic, the relatively smaller number who are, have ARC symptoms, and the still smaller number who have AIDS. Now, it's this most vulnerable population, the ones that are already infected, that attracted our attention. They're the ones whom we focused upon in an effort to see if we might be able to influence the course of events and hopefully, if possible, suspend the death sentence. Now, that approach we refer to as immunotherapy because these individuals are already infected. But those who are not yet part of that pyramid are those who are candidates for a vaccine to prevent them from becoming infected. And so when I hear the description of what we're doing now as the development of a vaccine, I want to make it clear that we're trying to develop is some way of treating those who are already infected, although we hope in the not-too-distant future to be able to turn our attention to those not yet infected. The next slide, please. Now, the properties of the virus that you've been listening to that make it special and different from any others and difficult to deal with, you've already heard, and you can read that yourself, that it gets right into a cell, not only into the cell, but it becomes integrated into the nucleic acid of the cell, into the genetic material of the cell, and from that point on, it remains to do its dirty work, so to speak, and has developed in the course of evolutionary time these marvelous strategies for survival in what would otherwise be a potentially hostile environment. So we talk about the war on AIDS, we're really speaking about a war between the immune system and the virus. And if you like, in due course, if we come to conquer this agent and develop some kind of, or enter into some kind of a negotiated settlement, some kind of a peace, we may find that the virus will remain, but the immune system may still be activated sufficiently to keep it at bay. The next slide, please. Now, I see this virus not as a simple virus infection, but as you'll notice in this triangular diagram, that it's more than a latent virus, it's an integrated virus, and it behaves more like a cancer and like autoimmune disease. It's awfully quite different from the common cold or from influenza or from polio, where you have a cell-free virus that destroys the virus and does its damage. But in this case, that nucleic acid, or the genetic material of the cell, continues to proliferate, increasingly with HIV code, and it's these cells that continue to replicate just as cancer cells are replicated. And that's for that reason that much of the treatment is like that of cancer chemotherapy. And now we're speaking about something analogous to cancer immunotherapy. And then you hear that there are immunopathic effects, that is to say, the immune system itself is intoxicated. Perhaps, if you like, there are toxins that are produced. Now, it's the investigation of all of these details that Dr. Gallo referred to, Dr. Fauci referred to, Dr. Rubenstein referred to, that are being worked out now in this remarkably elaborate social enterprise that I referred to as science. The next slide, please. Now, when you see what happens after infection over a period of time and the gyrations through which the antibody mechanism goes through, so the rise in antibody, and some fall away, some persist, it's in that period of time that we're hoping to introduce some kind of intervention. But there's only part of the story, because this tells you about antibodies and not about cells that attack cells. So antibodies, by and large, attack viruses that are in the free state, but it takes a cell to destroy a cell that's making a virus. The next slide, please. Now, this is the culprit itself. You see its complexity, and now you can see by the labels some of the things that were referred to, and we have opted in the immunogen that we have been working with to use the entire virus, but as it turned out, in the course of purification and concentration, those knobs, which are the GP120 knobs, the envelope portion of the virus, falls away. And inasmuch as we were intending to use it for immunotherapy, and since we have observed that people who are already infected continue to have antibody to GP120, we have decided to go ahead and incorporating this kind of a preparation, which has all of the viral proteins, not trying to second guess which one would be the important one, and combining it with some potent adjuvant or a potentiator to see to what extent the immune system can be stimulated, even though the individual's immune system is constantly being stimulated from within because of the constant production of virus. We have introduced it in an unnatural form, enveloped in mineral oil, for example, with other ingredients. The present for the purpose of attempting to trick the immune system into reacting even though it's already being overstimulated. And so the trick here is to see if we can do this at an appropriate time in the sequence. The next slide, please. And so you can see here schematically how we visualize it, this almost hourglass-like figure lying on its side. The curve that starts at the lower left-hand corner, that shows how virus-infected cells continue to proliferate. The one that starts in the upper left-hand corner, it shows how the defensive cells, or the cells of the immune system, diminish or deteriorate. And as you can see here, the objective is by immunotherapy and even in combination with chemotherapy to keep everything pushed to the left so as to maintain a favorable advantage to the immune system itself. The next slide, please. And so this is another way of showing the deterioration of the immune system. The antibody, the cells of the immune system deteriorate or diminish at a regular rate, and then all of a sudden there's the collapse of antibody, and in the next slide, and then there's one more after that, just to illustrate the strategy that we hope to employ, and that is, even in the later stages of the disease, perhaps to use something like gamma globulin produced against the HIV agent, together with active immunization to shift, or chemotherapy to shift, to restore the balance of the immune system. Now, I thought I'd show you this so that you can understand the strategy, and then the last slide puts this out in. Is there another slide? Yes. In some detail. And now you can see the master plan, so to speak, that we're hoping to unfold using the kind of a preparation I've referred to, and the portion to the right refers to the HIV seropositive individuals, all the way from the infant, then to those who are asymptomatic, and then those who are more advanced. And the objective there is to tailor a strategy suitable for the different stages of the infection. On the left-hand side, we refer to individuals who are not yet infected, and in the low risk category, we are contemplating attempting to produce antibody that could be useful on the right-hand side of the slide, and then, of course, in due time, after appropriate animal studies are done, and these very likely will be in the monkey, to use methods that have been demonstrated to be useful in the protecting monkeys from infection in human subjects. You can take the slides off now, please. And just by way of summary, so that you can see how the work is being orchestrated in a perfectly natural way, it's as if no stone is being left unturned. That work is being done all the way from the use of Dr. Goldstein's small synthetic peptide to the kinds of things that Dr. Gallo referred to and Dr. Rubenstein, to a much larger and more complex agent. And the assurance that this should provide is in the fact that so much work is being done over such a wide span of diversity that it will not be too long before we'll have the kinds of insights that will allow us then to focus down on what it is that might produce something useful in the nearest term future. Thank you.
Thank you, Dr. Salk. You've heard from 4 of America's leading experts on the AIDS crisis. We're now going to attempt some cross-fertilization. The time is short. The instructions are specific, so I'm going to read them, make sure that we get them straight. Please allow me to review the procedure. Each member of the media panel will have 3 minutes in which to ask questions of our panel of experts. and to hear their answers. Following the 4 members of our media panel will be the members of our advocacy panel. As you're aware, we do have a limited time, so please allow me to apologize in advance for interrupting your questions or answers at the end of the 3-minute opportunity. At the beginning of your question, please indicate to whom you're addressing the question. Thank you.
We've heard some things, as Mr. Anderson said, ranging from the very specific to the very general about this virus. There is one solution to the problem of AIDS that hasn't been brought up. It's either a very naive or a very far-out question, but I'm going to ask it anyway, perhaps of Dr. Salk, perhaps of Dr. Gallo. What would it take to rid the world of this virus, to make the virus go away? Could that be done? Has it been done in other diseases? Is the polio virus still out there in the world threatening us, and is it only because we are mostly vaccinated that we are protected? Or is the virus somehow changed as a result of your work and that of others so that the situation is different?
Polio is eradicable from the world and will be, just as is smallpox. It's just a matter of time. And I think the answer to your question about can it be done, the answer is we will see in time. But it's a far more difficult agent to eradicate from the human population. And also, it exists in a form in animal reservoirs which is not true of polio. And so my guess is that we may have to develop different strategies for keeping it at bay. And that very likely will be an immune defense. It will be different for those who are not yet infected, and that's going to be, I would say, relatively easy, even though others may question that. But I think that we will have the means for so doing. And those who are already infected, that's asking for a great deal to expect the virus to disappear from the host. And it may be, however, that that will require the attrition of the population of those who are infected.
Let me ask a question for anybody who cares to tackle it. Several of you are working on human vaccines in the early stages. Do you think that the situation in the United States and in other countries is as good as it could be to allow early testing of the vaccines? How rapidly are you able to proceed?
Well, when you say the situation, I think you have to think in terms of the state of the science. There's a reluctance, understandably so, to go into efficacy testing in man unless you've proven a vaccine candidate to be safe and effective in an animal model. That doesn't mean that phase 1 studies, I'm talking about in an uninfected population, that doesn't mean that phase 1 studies cannot be done, but there really is a dichotomy of opinion in this country and abroad as to how much do you need to know about safety and efficacy in an animal model before you move into a human, or can just immunogenicity and safety in a human allow you to move on to phase 2 or 3 trials. And I think resolution of that scientific question will really allow us to answer the question whether it's going to take many more years or just a couple of more years.
Where do you stand scientifically?
From a scientific standpoint, I think that you can learn a lot about immunogenicity and safety by going into humans in a product that has not yet been shown to be effective in an animal. Puristically, scientifically, I think you should do it that way, but I think there are populations throughout the world who may not be able to wait for that.
So Dr. Salk wants to make a comment. Let me ask if you would, in the process, clarify one small thing about the human vaccines. These vaccines are not all alike. Could you distinguish in just a phrase or two one of the potential candidate vaccines from another? The live virus for some of them?
No, live virus is absolutely out of the question. You've got to work with non-infectious agents. I would say that's generally true, but I shall not elaborate upon that. The use of an antigen that is going to produce the desired effect. Now, in the case of influenza, we knew what that was. It was neutralizing antibody. In the case of polio, we knew what that was. Not only neutralizing antibody, but simply immunologic memory. In the case of HIV, we've not yet identified the equivalent of that. If we were able to identify what the target is that needs to be achieved, whether it's neutralizing antibody or whether it's some other antibody or cell-mediated immunity, we would test for that. And then I would say it would justify going into the human population. But I think we will know that within the next short while from the simian studies, because they're being designed in that way.
Thank you very much. Our first questioner was Barbara Cullen.
One of the things that's so interesting in listening to all of you all discuss these vaccines is that you're talking about using all of the proteins in the virus, everything in Dr. Salk's virus going down to Dr. Goldstein's little peptides. And particularly, I would like to ask Dr. Ari Rubenstein, you're talking about yet another mechanism, hyperimmune, or not hyperimmune, but an antibody that is in the mother that prevents the virus from crossing the placenta. What is the condition of those mothers that have that antibody that protects their children? Are they in a different condition from their children or from one another, or what?
Well, first, we are not speaking about a different antibody. We are looking at neutralizing antibodies under various conditions. The mothers that we have studied, some of whom were in advanced stage of the disease and yet had these neutralizing antibodies. So that the presence of neutralizing antibodies does not necessarily indicate that that individual will have a salient disease or an asymptomatic disease. When we are talking about that vaccine, the PND vaccine, we have to understand also that we are dealing with a totally different vaccine that Dr. Salk is speaking about. Dr. Salk is talking about a vaccine to an individual who has not been infected by the AIDS virus. When we are dealing with infected mothers, these are individuals who have seen the AIDS virus, their immune system has seen this AIDS virus, has mounted some antibody response, and there may be a little piece of this antibody response missing that will determine whether the virus will be transmitted to the fetus or not. And we think that one of these little pieces, probably one out of many, are the antibodies, the high affinity antibodies that we have identified recently.
May I quickly correct the statement that what I'm talking about is a vaccine is what I'm using is in non-infected individuals, just the opposite. That was the whole point I wished to make, that we're working only in individuals who are infected. And that I call immunotherapy. And what Dr. Rubenstein is speaking about is in a way related to that, but it's a bit removed because it involves keeping the infant from being infected. But I want to make clear that I'm not speaking at this point about using that material as a vaccine until we have more information.
Yes, thank you.
Thank you, Ms. Fedner. Thank you, Ms. Fedner. Our next questioner is Dr. Susan Oki from the Washington Post. Dr. Oki.
I think maybe I should ask a question related to treatment, since we've talked about vaccines primarily so far. I get the flavor from the presentations that we've heard today that most of you are of the opinion that treating someone infected with the virus, it will probably not be possible to eliminate the virus entirely from that person's system, and that you're fighting a sort of war of attrition where you're using a combination of therapies to keep the virus at bay and preserve the immune system as far as possible. Since that seems to be the approach, I'd like to ask, has there been enough emphasis in research so far on other ways of treating patients other than attacking the virus's mechanism for replicating its RNA into DNA? One of the criticisms that I've heard of the treatment trials is that they've focused primarily on AZT and on other reverse transcriptase inhibitors. Are we doing enough in the way of research to treat AIDS-related infections and find new treatments for those, and to boost the activity of the immune system itself? And I guess I'd like to address that to Dr. Fauci, maybe Dr. Rubenstein.
Certainly we need to do more on opportunistic infections, both from a primary treatment standpoint as well as a prophylactic standpoint. The situation with opportunistic infections is that it's much more complicated to do treatment trials because people come into the hospital acutely ill, and it is much more difficult to get a clinical trial setting. Not impossible, it's just difficult to do. Early on in the developmental drug process, naturally, people focused on antiretroviral agents. Right now, over the last several months, 6 months or more, and for the next year or so, there will be a shift in emphasis so that the relative proportion of studies directed towards opportunistic infection, both prophylaxis and treatment, will be proportionally increased over those with antiretroviral. Reconstitution of the immune response is quite problematic in that it is very difficult, having already shown that bone marrow transplantation itself among between identical twins was unable to reconstitute the immune response. I think factors such as interleukin-2 and gamma interferon and others are being tested, but that's going to be a much more difficult situation than developing drugs for antiretroviral as well as for opportunistic infection. But the studies will be done. That doesn't mean that they're giving up.
Maybe I could ask Dr. Rubenstein to address the same question in children, and I'm very interested in particularly protecting infants, because presumably infants, if they're infected, are newly infected and maybe have not had the same damage to their immune systems.
Well, at the Albert Einstein College of Medicine, we have a so-called ACTG AIDS Cooperative Treatment Group. We are part of the National ACTG Network, and as such, we have access to a large number of treatments for adults and children with AIDS. One has to go forward very slowly, especially with immunopotentiating agents. And I just want to give an example of a treatment that we have attempted in 1982, and we're quite burnt badly with it. We have then shown that children with AIDS have a deficiency in thymic hormones very early in the disease course. And the results initially looked extremely well. Those kids who had multiple opportunistic infections improved significantly clinically and did very well for about 3 months on the treatment. And after 3 months, there was a sudden and rapid elimination of all their T4 cells. And we think what we have done with immunopotentiation there is that we, on one hand, boosted up their immunity, on the other hand, we accelerated the replication of the virus. So that antiviral therapy can go hand in hand with immunopotentiating therapy, but each one of them has to be investigated very carefully. We have access to more and more drugs to be used in children, and I believe that very soon there will be no drug used in adults that will not be used to treat children with AIDS.
Our final press panelist is Alfred Rosenfeld of Omni Longevity Magazine. Mr. Rosenfeld.
Thank you. It's Albert, but I don't feel the way about Alfred the way GW does about Georgetown. Good thing about, well, you said you couldn't pronounce all those things, right? Good thing about being last, I guess, is you can pretend you were going to ask all those bright questions that the people ahead of you did if they hadn't been first. One thing I want to bring up that hasn't been touched on yet is the question of the ethics of the experimentation. When Dr. Salk was working on polio in the 50s, school children could be inoculated en masse in very large field trials. I think we'd have a hard time doing that today. That was before the days of bioethics was thought of, before we had institutional review boards and so many strictures around doing experiments with people. I wonder if perhaps you, Dr. Fauci, since you represent the NIH, or anyone else who wants to comment, what are the specific difficulties now that we didn't have then, and how will you get around them in terms of finding populations?
If you're talking about vaccine trials, the difference between polio and what will happen, we don't have a vaccine yet, obviously, for HIV, is that in the polio situation, virtually every child was at risk. You're going to be talking about vaccinating at-risk populations, which is different from a disease that is behaviorally versus non-behaviorally transmitted. You would not want to, we can't say what will happen 50 years from now, but for example, if an effective and safe HIV vaccine were developed, you would not want to line up homogeneously everyone in the country and immunize them. You would have people who would most stand to gain from protection in certain behavioral categories or risk categories, be it here in the United States or internationally. So I think you'll have much, much more of an informed desire on the person's part to be immunized, because they may feel that they are in a certain risk of getting infected, as opposed to a respiratory-borne or other type-borne disease where there's universal homogeneous risk. So it's a much different situation than it is, for example, with polio.
Would it be easier to, why would anyone who doesn't have the infection, if you're going to test a preventive vaccine, a true vaccine,
right?
Perhaps, Dr. Salk, that's why you're working on the infected population first. If people know that you can stop the infection, then they might not be so afraid of the risk of getting it. Is that part of the strategy?
Yes, and if something could be done to ameliorate it, as has been suggested by immunotherapy and perhaps other means, then those who are perhaps infected will want to have themselves tested for early diagnosis, for early treatment. Clearly, that is the target population at the moment, and what Tony Fauci has just said is absolutely true. This would be like immunizing against cancer or multiple sclerosis. Who will be the candidate for such a vaccine if you, in fact, could prevent the initiation of the cancer or the multiple sclerosis? That's why it's much more a disease like a chronic disease that has to be treated. But in this instance, it certainly is going to be preventable. And the target population is, we know what the target population is in this country. In Africa, it will be everybody. And so I do believe that the strategy as used for polio will be necessary in Africa and other countries where the epidemiology is of that nature.
So one last quick question, if I may.
Here, I'm going to have to cut you off at this point.
Okay.
We're going to now have to turn to our advocacy panel in order to move this along. And our first questioner is the medical director of the Whitman Walker Clinic, Dr. Peter Hawley.
As a early participant and as a study subject myself in the 1950s polio vaccine, I'm going to address my first question about being a study subject. I think we all recognize the important efforts that NIH has given to making research protocols available to people who have been underrepresented so far in research studies, particularly women, children, intravenous drug users, people who don't have English as their primary language. But it would seem that largely because of the appalling poverty in much of this group of people, that any program is destined to not fully achieve the success of getting people in and through trials unless the issues of their full medical care are addressed. And few research studies worry about medical care beyond the study itself. Is there any possibility, and I guess I need to address this mostly to Dr. Fauci, is there any possibility of establishing a program that would help support the care for those people who are simply unable to provide their basic medical care from their own resources?
That's a very important problem that you mentioned, Peter. What can be done from the standpoint of the NIH would be to draw up, as we have, collaborative efforts with other agencies, for example, the Public Health Service, to have the research resources be used for the delivery of health care. I think you would agree would not be a good way to spend research resources, but what we need to do is to get resources for health care delivery incorporated into the whole package. And in that regard, We've already initiated demonstration projects with HRSA to do that. But as you are well aware, a demonstration project is a demonstration project. It doesn't really attack the picture as it exists in society. So what we're really left with is what Admiral Watkins said when he made his summary of the President's Commission, that HIV has really forced us to look at so many of the terrible inadequacies in our society, particularly the healthcare delivery system. But the best that one can do is to try and coordinate agency efforts in that regard.
We've heard a lot so far from various people about humoral immunity and how humoral immunity can be potentially used to alter disease in people, but rather surprisingly, from an immunology-oriented discussion, very little on cell-mediated immunity. Would some people, maybe Dr. Goldstein, like to address other areas, particularly cell-mediated immunity, that could be harnessed?
Well, I think what Dr. Salk indicated in terms of capsuling the various vaccine approaches from a subunit vaccine, a core vaccine that we're working with, to a killed core vaccine, to the envelope vaccines. I think the ability of any of these vaccines to elicit T-cell responses, cytotoxic killer T-cell responses, are going to play a component, if not a significant component, in whatever will be the final candidate vaccine that proves to have efficacy. And that may include pieces of the envelope, or as well as one or more pieces of the core protein. I think Bob Gallo referred to that approach that is being carried out by Daniel Zaguri in Paris. In our own studies, we have found that HGP30, the P17 region, is a very good inducer of T-cell cytotoxic responses. And whether that response, which should translate perhaps into eliminating infected cells, translates into an effective vaccine in a vaccine setting, remains to be established.
Thank you. Our next questioner is Sandra McDonald, chairperson of the National Minority AIDS Council.
We've heard some discussion today about the underserved and the other phase, or the phases of AIDS that we're now seeing. Could you give some thought to ensuring that this underserved patient, who we're not seeing now in clinical trial, who will not be able to afford AZT and therefore is not taking it, would be able to participate in new drugs and new studies? And certainly when we get to the point of a vaccination, how do we ensure that we will be able to have these people involved when we get to that point? Secondly, has there been any thought to looking at addiction? We do know that many of these patients are addicted, and no matter what the substance is, there is addiction involved. Has there been any correlation of addiction behavior as it relates to the research of HIV and how we can help those two things go along together? And that's just an open question.
Well, from the standpoint of getting underserved populations into clinical trials, certainly the relative proportion of individuals with HIV infection of color and minorities, others are really much less than in the clinical trials than those people who are infected. The way to get them into clinical trials is to put pressures on the clinical trial programs to enroll minorities, and the way we've done that is to tack on added contracts to the clinical trial programs for an incentive to bring minorities into the programs. We have a program with NIDA, the National Institute of Drug Abuse, so that they will collaborate with the NIH on clinical trials and provide the incentive of granting extra amounts of money for people who bring in IV drug users into the program. Right now, 10% of our clinical trial participants are IV drug users. That's less than the more than 20% of AIDS patients who are IV drug users. So we need to bring that number up. If you bring that number up, you will bring the minority up, since the majority of the IV drug users with HIV infection are minorities. So the way to do it is to link support with the incentives of investigators to get minorities into trial. And we are seeing an increase. Over the last 18 months, there has been a steady increase in blacks and Hispanics in the program. It's still below the target of what it should be. With regard to the broad problems of healthcare delivery, this is of such a serious nature that it just goes right into the whole problem of inadequacy of healthcare delivery system, which Secretary Sullivan is trying to address as best as he can.
Dr. Rubenstein, I'm interested in what you've been able to do in New York. Certainly, we know that you do have the numbers of women and children. Interestingly enough, early on, we thought that many more infants would be HIV positive when born to these women, and now we see that is not happening. You talked about the antibody. I'm not clear, are you making it available to patients who are already infected as they go through, or are you in a trial setting? Is this something that will be widely available for other patients? Where are you now?
Well, the majority of our patients belong to minorities. Over 90% of the patients at the Albert Einstein College of Medicine are minorities. The antibodies that I've been talking about, the high affinity antibodies, are not available yet for trials in humans. They will be available for primarily the minority populations in our area, because this is a population we are dealing with. For example, when I'm talking about a transmission-blocking vaccine in pregnant women, in fact, our target population are drug-abusing women in a methadone maintenance program. We know, for example, in one of our methadone maintenance program that we will see about 180 to 200 pregnancies a year among those HIV-infected women. These will be the first candidates for the vaccine or for an hyperimmune serum.
Our next questioner is Michael Murdian, the Executive Director of the National Association of People with AIDS.
Dr. Fauci, you've talked about the shift in emphasis that NIH is taking, and I know all these questions are being directed at you, but can you talk about the dollars and the percentages of dollars that are going into antiviral research versus the opportunistic infections?
I don't think you could put a dollar value on it. You can talk about the numbers of people who are in trial and the relative number of protocols. For example, the numbers of protocols that had opportunistic infection has grown in the last 12 months from 10% of the protocols to 20% of the protocols. The number of people who are on protocols that for opportunistic infections have grown from 600 to about 2,000. For the next 12-month planning of the protocols that will be implemented from now until the summer of 1991, there are 5 new HIV or 6 new HIV and 12 new opportunistic infections. So there is a shifting of the balance with the goal and the target to be somewhere around 30 to 40% of all the protocols will be for opportunistic infections. Now, it's difficult to put a dollar value on that because it depends on how many patients you have and the kinds of questions you're going to ask. I would imagine that when it comes down to it, the dollar figure will be much more expensive for opportunistic infections. One of the problems we face, and this is a problem that's more of a sociological healthcare delivery problem, is that if you're studying someone in clinic who's a relatively asymptomatic HIV-positive individual and you give them AZT, that doesn't cost much with regard towards the care of the patient. If someone comes in the hospital through the emergency room with toxoplasma encephalitis or cryptococcal disease, if they come into the hospital, as soon as the hospital finds out that you want to do a protocol study on them, many hospitals will immediately withdraw the third-party payment, so that all of a sudden the protocol has to pay for it. So instead of costing $500 to $1,000 to do a patient, it's going to cost $15,000, $20,000 depending on how sick the person is. This is something that needs to be addressed that's beyond our control.
A quick follow-up to that. Are you involving other doctors in this, particularly in dermatological aspects of the development of HIV?
Yeah, we have collaborative efforts with the National Institute of Arthritis and Metabolic Disease, which is responsible for the cutaneous manifestations, and we're working with them and trying to develop joint protocols with them and joint basic science approaches.
Okay. This question is to Dr. Bernstein, and there's a lot of talk among my constituency group about the funding that's being generated for pediatric AIDS research. And to many people living with AIDS, this money seems disproportionate when these drugs will be developed and tested in adults, and then they will be used in babies and children after being adjusted for the weight and size of the child. I have a difficult time in answering this question. I'm sure it's a question that comes up for many people here. And you're saying that there's more and more access to drugs. Will you please explain to us just what is pediatric AIDS research?
Well, that's quite a broad question. First, I do not believe that there is a disproportionate funding for pediatric AIDS as compared to adult AIDS. Many people, I guess that's what you meant by saying disproportionate, that too much money is spent on pediatric AIDS.
When the drugs are being developed in adults and just proportion for size and weight of the child?
Well, again, I can't put a dollar number on the amount used for research in pediatric AIDS and in adult AIDS. Many people don't realize what the numbers are in pediatric AIDS. For example, in the Bronx, we have created the Bronx AIDS Consortium. We have just created now a list of HIV-infected children in the Bronx alone, an unduplicated list, which came to close to 700 children. There have been several studies conducted in the Bronx looking at the incidence or prevalence of HIV positivity in pregnant women, which in some hospitals goes between 5 and 12%. So that we expect annually to see about 750 to 1,000 new babies infected by the AIDS virus. So that the population of children with AIDS is unfortunately increasing very rapidly. The intensity with which one has to approach an infected child, I believe, is much more than the one needed for the adult patient with AIDS, especially because most of these patients come from minority groups, they're underserved, and a tremendous effort has to be placed into reaching out to these patients.
Thank you very much. Our final questioner on the advocacy panel, B.J. Stiles, President of the National Leadership Coalition on AIDS, please.
In the 10 years we've been dealing with AIDS, we have never lived in a time of quietness or lack of disruption in response to the epidemic. Conflict and disagreement and often acrimony characterize the worlds that you live in and that both the journalists and the activists deal with. Because of their singularity of experiences, my first question is to both Drs. Gallo and Dr. Fauci. In concluding, if you were commending the press for having done an outstanding role in this epidemic thus far, what one or two things would you compliment the press for? And looking at the next 10 years, what one or two things would you most urge the press to do better or differently? And if you were also complimenting activists, would you have a compliment to give? And what would be your strongest criticism or suggestion for the future? Dr. Gallo?
Well, that's a good question. It's a good question. I do think you wasted your opportunity. I could better comment on a number of the scientific questions that came up. I don't want to be an expert on the media, but I think above all what the media needs is what one of the editors of Le Figaro, of all places, said to me a few weeks ago. An international committee composed of ethical standard reporters combined with some scientists to know how to comment on immediate claims, immediate articles, so that there wouldn't be the automatic follow that there is when the tail is being chased by people in the press. When an article comes out, I get the opinion that there's an automatic machine in the press, that it just keeps running with no real question in detail, and that's because in a society that we live, which is a capitalistic society, you have to sell the next day's newspaper. So I think the idea of an ethical committee of high-level people in the press from several sides of the world, along with, let's say, some kind of scientific advisory panel, could stop a lot of the things that weren't stopped in miscommunication. Whether it was HPA 23 and Rock Hudson, which was a mistake, whether it was mosquitoes, cockroaches, cyclophosphamide, or whether it was sometimes things that relate to history of the field. A lot of it could have been avoided. Regarding activists, I'm, again, not an expert, and I haven't followed it so carefully. I think the positive side, I should have said the positive side of the press is I surely think they've been interested. And and wherever there are science writers, there have been, I think, accurate, fair, and good reporting in general. Among the science writers in the press are those that have really followed things carefully for some years. Activists, I think the number one achievement has been to make us think practical all the time, not just when it's obvious, but to keep reminding people in the laboratory that every time you have an observation, think what you could do with it today, not in a year or two when it's really grossly obvious. In the negative sense, I think it's very frequently misdirected arrows, almost pathetically so.
Dr. Salk, there apparently are close to 1 million Americans who are HIV presumably asymptomatic. If you had a grandchild who was in his or her late 20s who look at the next 10 years of the epidemic, what counsel would you have for them, and how would you hope they would behave?
Well, inasmuch as we know how the HIV is transmitted, and inasmuch as this is associated with certain forms of behavior, that those forms of behavior should either be avoided or engaged in with the appropriate precautions. The difference between what was true 35 years ago and now is that one didn't know how poliovirus was transmitted or when one would encounter the virus. It was ubiquitous in the population. This, on the other hand, is quite well restricted. You really have to try hard to become HIV positive. It's not something that comes to you easily.
We want to thank our panelists for their probing questions and our 5 distinguished experts for their thoughtful and informative answers. Now we have the opportunity for you in the audience to participate in this program and to address questions to these most distinguished experts. We have a limited time. We have a strict procedure. If you'll line up at the microphone in the middle aisle, one of our people will assist you. We appreciate, because of the shortage of time, we'd appreciate it if you'd limit your questions to 1 and that you would address your questions to the distinguished experts. Please.
My name is Jeff Levy. I'm going to turn the tables just a little bit with a very quick question. One of the fundamental tenets of medical care and medical research in this country is a respect for the confidentiality of a patient's medical records as well as research records. Without those confidentiality protections, it is felt it would be impossible to get people to come forward for either research protocols or for treatment, particularly when we're dealing with a disease like AIDS where people tend to be stigmatized. Mr. Anderson, in 1985, you published in your syndicated column material from the medical records at NIH of one Roy Cohen. While many of us may find his politics to be reprehensible, the principle remains that this was a gross violation of patient confidentiality. Given that we're sitting here today talking about research, I was wondering, Mr. Anderson, if you could tell us whether you considered the impact on the ability of these gentlemen in front of us to recruit for their clinical trials and research efforts when you published that information, and perhaps turn the tables a little bit as well in asking Susan Oki, who is both a physician and a reporter, to see how she might balance those issues, and finally ask Dr. Fauci to tell us whether this kind of publicity or publication has a negative impact on his ability to recruit in the NIH clinical trials?
Well, we'll make an exception. The other questions, we'd appreciate if rather than tear me apart, that you'd tear the members of the panel apart. That would be wonderful. The reason that we wrote the story about Roy Cohn was not because of his political views, but because he was infected, because people at the NIH brought us records that he was still soliciting sexual intercourse with people without notifying them that he was infected. They were outraged at this. They felt that the public interest was better served by exposing the fact that he had AIDS. These were the people who brought us his medical files. We did deliberate, we did agonize over whether to publish them. Finally decided that it was in the public interest to do so. That's the reason we did. If anyone wishes to disagree, please feel free.
I've had to deal with these kinds of issues, I would say more often when it comes to illnesses of heads of state or presidents. I think that the public has some right to medical knowledge when the president is ill and when the illness has something to do with his ability to perform his duties. But in this case, I don't think that I would have done it.
The effect on the clinical programs was quite negative. Several of our patients, when they saw that printed in the newspapers, were very concerned as to their own confidentiality. I don't have any idea how that... Mr. Cohn was not one of my patients, was a patient on the Cancer Institute ward, and I don't have any idea how that record got out, but I can tell you from the patients who were on the clinical trials, it created a great deal of anxiety as to whether or not their confidentiality would be kept, and on the whole, it was a setback for us with recruiting people into clinical trials.
My name is Terry Beswick. My question is for Dr. Fauci and Dr. Gallo. And it's in regards to well, living in a capitalist society, we have to recognize that there's a marriage between private industry and the government that is in many cases productive towards coming up with effective treatments, we hope. However, my question is, do you feel that there are times when there is a problem, an ethical dilemma for individuals who are essentially public servants or institutions that are funded by tax dollars in collaborating with private industry and causing such things as the KS drug that Dr. Gallo is referring to, the TAT drug? Is that so secretly being researched right now with pharmaceuticals, and we have an NCI employee funded by tax dollars who's cooperating in keeping this drug secretive? And we understand there's privatization, there's protectionism and this kind of thing, but given what we've seen before, and with all due respect, Dr. Gallo is the co-discoverer of the virus, I think we can recognize that there's times when conflicts of interest can come up and problems and lack of accountability to the public.
Yeah. I think it's a fair question, and it's sometimes an important question, but believe me that there's a lot of miscommunication. And some of the worst problem would be if there wasn't respect for confidentiality related to the drug. There's so many rumors about the Kaposi sarcoma drug because there's people working on Kaposi sarcoma in collaboration with drug companies of more than one kind. The drug that you're referring to, if it refers to me, came out of the systems we developed at NCI, in part to a major degree by the very individual that you're speaking of. That was the invention from the National Cancer Institute and from American taxpayers' money, the system to study Kaposi sarcoma. It was our idea, the idea from myself and from my two co-workers, to go to a certain category of compounds to see if it would work in the system we developed. So the system was ours, development. The idea for the drug was our idea. We looked to see where we could get this category of drugs. It's a Japanese pharmaceutical company. We contacted that firm. We obtained this category of compounds, and we did experiments that we think are interesting. We suspect they could be important. Nobody in this world has to tell me to move faster than I want to move. I feel sufficiently competitive and sufficiently qualified to move something that we discover. It's not that we want to hold something back. There is no gain, certainly none financially. There's certainly no gain for us competitively. There's certainly never have I ever wanted to slow down. My problem is from going too fast through my career. In any case, in order to have that program go forward further, we contacted this company and met with them. They had never collaborated with the United States government before. They never collaborated with any government. They never collaborated with academia, only within pharmaceutical companies within Europe and in Japan. They wanted a confidentiality agreement to get 1 iota more of the drug for us to study. We had only the first stage of the study completed. We signed a confidentiality agreement legally and properly to be able to get more drug to be able to complete the studies. They are interested now in forming a collaborative research agreement with NIH, with my laboratory. We are in the process of signing that. The director of the company is coming to visit me in mid-July. Any publicity related to this compound will shut off the collaboration. It'll kill it. And one of the points I was referring to of no, not what the compound is. You don't know. What I did was answer questions related to the compound in an honest and open way at a meeting that 1 or 2 people attended and then wrote about it in an excessive, exaggerated, and ridiculous way in the ridiculous newspaper in New York. That set off a chain reaction. You understand? So what I did was presented our data on Kaposi sarcoma and answered a question if we were testing drugs and said yes, and we had some interesting and opening data. But I never mentioned that drug to this date, and you don't know what that drug is, and your speculations are nonsense even in that paper. So the stage that this is at now is trying to move it as fast as humanly possible with collaborations with the Japanese pharmaceutical company, and our relations are extremely good up to this point in time that I speak to you. I want to maintain them that way so we could get the drug as fast as possible through the FDA and properly test it. I know of no other rational way of dealing with the question. Regarding industry relationships, the formation of collaborations with industry is tricky. You want to move a problem as fast as possible. Some people, not doing so well in their personal lives, sometimes got tempted to be too close to industry. That happens, it'll probably happen again. It hasn't happened with the vast majority of people that relate to industry. Everybody that's sitting up here relates to industry. Some have their own company. Some collaborate with companies. It is an impossible problem to solve without dealing closely and collaboratively with industry. But there's no Japanese-American relationship that requires us to keep secret so that we can get some funding to our laboratory or funding personally. That's just nonsense.
My name is George Rainey, and I would like to address this question to Dr. Rubenstein and Dr. Fauci regarding the United States' current ban on fetal tissue research and how that affects AIDS research currently, since it was enacted by, I think correctly, the Reagan and Bush administrations. And also, Dr. Saut, could you clarify what, and earlier when you were saying about attrition of HIV populations and treatment of people who are asymptomatic and those are people who are not HIV infected and people who are HIV infected. Was it attrition of the various opportunistic infections or attrition of the populations?
I
think you were talking about attrition of the population.
Yes, of the population. That in due
clarify
course,
what that means.
In due course, if there are no newly infected individuals, then in due course those infected would diminish in the population.
Okay. And if you could continue with my first
Think of it
question.
in terms of population dynamics.
Right.
There's not a ban on fetal tissue research at all sponsored by federal funding. There is a moratorium on fetal research from induced abortions for the purposes of transplantation. That's what it is. So direct answer to your question, is that having impact on AIDS research? Right now, no. But if in fact it's demonstrated that fetal tissue transplants might in fact benefit an HIV-infected individual, and we were not allowed to do research for transplantation purposes, then a scenario might arise where that would be a problem. But right now, we can do in vitro test tube fetal tissue research. Many of us use fetal tissues for cell lines. In fact, a number of laboratories at the NIH and in NIH-funded institutions. But the moratorium you're talking about is with regard to transplantation. It's conceivable that that could be a problem. Right now, it isn't.
Hi, my name is Alex Warren. I'd like to address this to Dr. Gallows. This is something that hasn't been talked about, and it is the accuracy of the test, the antibody test that you developed. Do you consider that to be 100% accurate? If not, are there other tests that are available to detect the virus? And is it still considered that it takes about 6 months to appear in the blood system?
Well, to answer the last first, it's not 6 months. That's very late. It's more like a few months. But it can be as long as 6 months and sometimes a year in very exceptional cases. No medical test is 100% certain, nor is this. Done properly, and although the ELISA was mentioned many times, what I believe our contribution more was to emphasize, well, the blood test, yes, but to emphasize the need for confirmation by Western blot. That was the introduction of the Western blot into clinical medicine, was the development of the blood test for HIV. With the Western blot done properly after an ELISA, it's pretty rare and pretty stupid to make a false positive. A false negative will always occur because the simple reason is sometimes antibody levels are too low or appear too late, as you were getting at. So some slip by to this day, but it's a very small number. Yes, there are other tests looking for the proteins of the virus instead of antibodies to it. That test is also not sufficiently sensitive, in our opinion, to be the final answer. There's also the technique of looking for viral genetic information called the polymerase chain reaction, or PCR. That technique is not widely enough available to be the answer, but there is, of course, more work in this direction. Suffice it to say, it's an extremely good test, and there's room for improvement.
My name is Jeff Nelson. Dr. Fauci, earlier you were discussing inoculation, and you stated that with polio, every child would be at risk. But because HIV is transmitted behaviorally, the impression I got was that because it was a behavioral situation, there wasn't going to be such a large population at risk. It is my understanding that sexual activity is a pretty universal thing. So I guess my question is, what is the NIH doing about education about safe sex practices? And also, I have one comment for you, Dr. Salk. Excuse me. With all due respect, I think you can probably use the word condom in this room, and if I had a grandchild that I would want to counsel, I would say, use a condom.
The NIH's mandate is not one to be educating on behavioral research. That's just not the mandate. That's much more of the CDC. So what are we doing about education regarding safe sex? The only time we get into that is in some of our sexually transmitted disease research centers, but it's a relatively small effort. The major bulk of safe sex education is predominantly the CDC. There is some outreach behavioral research also in youngsters and adolescents and among mothers concerning HIV infection from the Child Health Institute. Getting back to what I was saying about homogeneously distributed infection is I still stand by it, and I think if you think a moment, I was not referring at all to the fact that sex is not something that's universal, but if you have an influenza epidemic, it can kill a 85-year-old monogamous couple, heterosexual. It is very unlikely that an uninfected, monogamous, heterosexual 80-year-old couple are going to get infected with HIV. So that's what I mean about homogeneously throughout the population. That's not making anything judgmental at all about any kind of behavior we're talking about, but the epidemiological facts are that this infection will not be homogeneously spread throughout the population. So you should focus your attention on people who might be exposed at a higher risk than an absolutely infinitesimally small risk. That's what I was referring to. Nothing judgmental about anybody's behavior.
I apologize. Time is slipping away. We have time for only 2 more questions. I apologize to those in line who won't be able to ask their questions.
My name is Patrick Donegan. I'd like to direct this question to Dr. Fauci. One rapidly growing population of women or people infected with HIV are pregnant women, and they, by and large, have been excluded from treatment trial participation. I'd like to get your thoughts about how high a priority protocol development for pregnant women should be.
It's a very high priority. In fact, there is a protocol now treating infected women in their 3rd trimester of pregnancy with HIV. As you can imagine, there is concern, safety concerns,
AZT.
both... Excuse me.
Not HIV.
No, I said AZT, didn't I? Excuse me. Obviously, I meant AZT. If the safety concerns, both from a teratogenic effect as well as from the effect on the mother, are obvious, but it's very clear that the risk to the mother herself of being infected and delivering, and to the child, we know is 30 to 40%. So there is a protocol, and will continue to be protocols, looking specifically at women in the 3rd trimester. You would hesitate to give a drug such as AZT in the 1st trimester of pregnancy. The difficulty is that now studies on looking at tissue from fetuses that were aborted in the 1st trimester and studying placental tissue indicates that the infection can be transmitted as early as the 2nd and 1st trimester, although most of it is probably at the area of, around the level of the 3rd trimester. But there are clinical protocols right now looking at just that, as well as clinical protocols for perinatal transmission, giving AZT to infants upon birth to determine if you could make that 30 to 40% infection rate go down to 2 or 3 or 4 or 5%. So it is a high priority.
Good afternoon. My name is Gerald Askew, and I'm here from the University of Texas of Dallas, and I was invited today, and I'd like to thank you for having me here. We have a problem in this country and in this world, and I'd like to challenge each of you on the platform that if we could we form a consortium of you as advisors, as the advisory committee, and if we have to give all of you an honorary doctorate so you can speak to each other on the same level, would you form a consortium so that we can address the issues from both a medical pharmacology, a pharmaceutical approach, a healthcare approach, a provider approach, and a religious approach? Can we come together as a nation and as a universe and address this together? And can we implement a system by which we can raise funds to do this? We have a 1040 form on our taxes that asks you if you want to donate $1 to the deficit of the United States. Can we put on that tax form, would you donate $1 to the research and distribution of services for AIDS victims and research for the prevention of AIDS? Can we put on your legislators' cards? Can we ask your states, your senators, your congressmen to give 1% of revenue sharing dollars that are tax-based back to the community for services to and services for AIDS victims, for food, for shelter, for clothing, and above all, for direct services? Thank you.
Speak to the issue, all right.
Well, thank you very much. I'm sorry. I do apologize to those who didn't get a chance to answer the questions, to ask their questions, but we are running behind time. We've heard some conflicting views about AIDS, but one thing is certain. Regardless of the controversies, regardless of the politics, regardless of the astounding scientific breakthroughs in dealing with the disease, AIDS has touched each one of us in a personal, a profound, and lasting way. And now, before we close, we have one final thought.
Obviously, the audio portion of the tape is not being heard. Thank you very much for attending and your support for this conference.
Keep the love alive, keep the will alive, keep the hope that time will heal the wound. Hold on through the night, you
That concludes this forum on AIDS and the challenge of the 90s. We invite you to join us Tuesday morning at 9:30 Eastern Time for live coverage of a House Appropriations Subcommittee hearing concerning funding for the National Endowment for the Arts. Coming up next, Senate debate on the AIDS Care Bill.
Thank you.